Noswaith a croeso. Good evening and welcome everyone to this evening's lecture to be given by Dr. Dayne Beccano-Kelly from Cardiff University. So Dayne completed his undergraduate degree and PhD in the University of Leeds, both of which included an educational stroke professional placement at the Mayo Clinic in Jacksonville, Florida. Dayne subsequently went on to do postdoctoral placements at the universities of Dundee, British Columbia and Oxford. In 2021, Dayne was awarded a UKRI Future Leadership Fellowship to start a program of research in Cardiff University on synaptic health in disease. Over the last 17 years, Dayne has studied a broad range of neurodegenerative conditions, including atypical Parkinsonism, Alzheimer's disease, and Parkinson's disease. His target is to recapitulate the progressive nature of Parkinson's disease in order to identify ways in which to intervene with the disease process. Dayne is an advocate for underrepresented groups in STEM and is a member of the Race Equality in Research Steering Group for Parkinson's UK and the Equality and Diversity Task Force for the Physiological Society. He has helped to initiate the Aspiring Black Physiologist Award to encourage Black students to pursue a career in science and has done a number of articles and press releases on the subject. He is a member of a number of committees, including the MRC Infrastructure and Capital Strategic Advisory Group and the Conference Committee for the Physiological Society. It's a great pleasure to have you here this evening, Dayne, and we're very much looking forward to your presentation.
Thank you very much for that very kind introduction and indeed for the invitation to speak to you all today. It really is an honor to be asked to present the work in the field as it stands and especially about the work that I'm trying to establish within my own group here at Cardiff University at the Dementia Research Institute. I think it's really quite vitally important that we try to share the data that we do as scientists with the people that it affects. And with Parkinson's disease, because it's such a global effector, it's really important to share it with everybody, to try and show what we're doing in order to try and combat the disease. So what I'm hopefully going to go through with you today is hopefully illustrated by this graphic here. It's something that I think is fantastic in nature. It's this ability for the cells in the brain to communicate with one another. With the chemical electrical activity that occurs in the brain, this communication between these neurons within the brain, the cells of the brain, really allows us to do everything that makes us us, from us being able to walk and talk to us being able to think and reason. However, this highly energy demanding state actually might be a double-edged sword. Whilst it allows us to do all these fantastic things, it may also open the door to degeneration at large, and the degeneration that we see in Parkinson's disease. And it's this that we really want to understand. How is this communication between the neurons of the brain actually occurring, and how is this miscommunication leading to Parkinson's disease? How then is it also contributing to the initiation of the disease, and when is it actually occurring? With these in mind, what we hope to actually be able to do is to develop better and better therapeutics that can hopefully one day halt the progression of Parkinson's disease. So Parkinson's disease was first described by its namesake, James Parkinson, in 1817 in his essay on the shaking palsy. It's a disorder that is commonly associated with old age, with the clinical symptoms often manifesting at around 65 years old. Now, what I hope from this lecture, by the end of this lecture, we take away is that we should possibly stop thinking about this disorder as being a disorder of old age and more about a disorder of aging in general. The manifestation, the clinical manifestation, and the associated symptoms that are often thought of when we talk about Parkinson's disease is normally these motor functions, this rigidity, this stooped posture, and the masked facial expression that you can see with Parkinson's disease. It's also accompanied by that hallmark, that tremor, that shaking that is so indicative in our minds of Parkinson's disease. Now, within the brain, war is being waged. There are a number of proteins that stop functioning as they ought to, and one in particular, known as alpha-synuclein, begins to start to stick together. And as it sticks together, it starts to form these clumps within the cells of the brain. And this is a signpost for us to understand that what we're dealing with is actually Parkinson's. These clumps are known as Levy bodies, and they are hallmark to the disease and are named after Fritz Levy, who first identified them. We actually get destruction and cell death of certain cells within the brain. In particular, one of those that is particularly affected are those of the substantia nigra pars compacta. That literally means the black substance that's stuck together, and actually in this particular piece of brain tissue, we can see them as this sort of jet black line. It's almost as if somebody's taken a marker to the tissue and has drawn a line on. And these particular cells, these particular neurons, release a chemical known as dopamine. Now, dopamine is important in a number of functions, but one of those functions is driving coordinated movement. And so the loss of these contributes to the motor symptoms that we see. By the late stages of Parkinson's disease, we can see that there's a substantial loss of these particular neurons. Now, despite decades of very intensive and fantastic research, which has led us to understand a number of the key players in Parkinson's disease, we still have no known interventive therapeutic that can completely halt the progression of Parkinson's disease. And I believe this is due in part to the focus on the very late stages of the disorder, the end stages where we see cell death. So how can we move forwards? What can we do to start to develop more and more therapeutics that can hopefully one day combat and intervene in Parkinson's disease progression? Well, from the information that we have so far, one of the things that we can start to draw up is a timeline of the disease. At the point of diagnosis, at about 65 years old, on average, we start to see that there are the manifestation of motor symptoms shown here in this red line. However, what we can see is that this green line, which symbolizes non-motor functions, starts years before those motor functions that are actually associated typically with Parkinson's disease. These are functions or dysfunctions such as sleep dysfunction or sleep disturbances, as well as impairment of problem-solving skills or chronic pain or anxiety. You can also see that those neurons that are releasing that specific chemical dopamine, those dopaminergic neurons that are found within the brain and that are destroyed within the brain in Parkinson's disease, are already 60 to 80% lost by the time we reach diagnosis. This means that there is years and even decades of cell loss that precedes the diagnosis. But with this information in hand, we can begin to identify earlier and earlier stages at which we might intervene and slow down the process of Parkinson's disease. But one of the things to really to hold on to and to understand is that the neurons of the brain do not just die simply just like that, and that there's a period of time that precedes this cellular destruction that actually leads to the cellular dysfunction. What we see here is there's a period of time that the cells don't operate as they should, and this dysfunction leads to the degeneration that's seen in Parkinson's disease. So if we understand what's going on here, we can essentially, hopefully, stop it from ever reaching the point at which cells die in the first place, and could hopefully slow or even stop the onset of Parkinson's disease. So in order to do this, what we need to do is to draw up a timeline of the dysfunction, a timeline of this particular phase of the disorder. And what I want to do in my work is to try and generate the understanding of what's going wrong before the cells die. And I liken it unto a car engine going wrong. There are a number of different things that might go wrong and might show up before we actually see the car engine break down. We might hear a rattling sound. We might see the check battery light come on. We might see corrosion on the car battery. But if we start to understand exactly what's going on, and very importantly, when it's going wrong, we can start to understand when we should take it to the mechanics in order to get it fixed before we actually break down on the side of the road. If we replace all of these different functions and indeed dysfunctions with roles within the cell, we can understand that if we can understand and tackle the dysfunction that leads up to the degeneration, we can stop the cells from dying in the first place and slow the progression of Parkinson's disease. So what should we be focusing on? Well, as we've mentioned, it is neurodegeneration. So the neurons of the body and of the brain are the ones that are specifically being attacked. So it stands to reason that there must be a particular function of neurons that is specifically making them vulnerable. Now, neurons of the brain are fantastic at this chemical electrical communication. It's what makes them special and unique and is their main role within the body. This is a neuron within tissue. We've managed to put into it a dye effectively to allow us to see this neuron in all its glory. And if you see the main cellular body that we have here, we can actually see its arms stretching out to reach out to make other connections, a bit like an octopus or a spider. It's actually reaching out to form connections with other neurons, a bit like us during COVID. It's absolutely desperate for contact and for communication. And when we look at these projections, we can see, if we zoom in, that they are actually a little bit knobbly. They have these little structures on the end, and each of these structures are known as boutons. Each neuron has several hundreds of thousands of these, and they are both the ears and the mouths of the neuron. They contribute to enable them to be able to communicate with other neurons. And so what normally happens is that you get an electrical impulse activity move down these particular arms and reach these boutons. And when it does so, it actually releases chemicals when it reaches this structure. These chemicals move across this gap and reach the other end of a bouton on another neuron, and then are changed and back into electrical activity. Now, this entire structure is known as a synapse, Greek for junction, and it's these that allow the crosstalk between neurons. And this communication is vital to executing actions within the brain. In order to keep this communication intact and to actually allow the execution of these functions, we need it to be like any other conversation. We need two things that are critical in order to be able to act on this. The first is to speak accurately so that the execution and the instructions are understood well. The other thing is to hear it correctly. We need to take the headphones off in order to listen to the person that's talking. And in this case, for the synapse, one synapse to talk to one another. So what we have here is when this is working in unison, when it's being spoken clearly, when it's being heard correctly, we can execute all of the specialized functions that make us special, the thinking, the speaking, and the movement. However, if we have a breakdown of one or t'other or both of these functions, then we will lead to miscommunication and indeed an inaccurate response to some kind of stimulus. So now we know what we want to look for. We want to look for any changes to the communication between the neurons. But how are we going to do this? Well, what we have is a conversation between two people, between two neurons. And if we imagine it between two people on a telephone line, we're able to use technology to tap into that telephone line. And in much the same vein, we can use technology to tap into the conversation between two neurons. We use something called electrophysiology, and specifically a piece of equipment known as an electrophysiological rig. This is a picture of one we have in the Dementia Research Institute, and it can look quite daunting and perhaps a little complicated, but if we actually break it down, it's much less so. We have a microscope that allows us to see these tiny cells, which are normally housed on this little white dish and this little white bath here. We have pumping systems to pump solutions over the cells to keep them happy. We have cameras so that we can visualize the cells on a larger screen. We have the ability to move mechanical arms down close to the cells in order to record from them the electrical activity that's going on. We have a computer that allows us to receive and interpret said information, as well as relay information back to the cells to see how they respond. But perhaps even more easy to understand is the fact that all of this boils down to being an open circuit into which we can plug our neuron. It's much like taking a bulb and screwing it into the socket in the ceiling. we can now control that bulb by switching on and off the light switch, and we can tell how quickly the light bulb comes on or how bright that light bulb is. In much the same way, we can plug in our neuron and we can tell a lot from actually altering the input into that system and listening how it speaks back to us. In a little more detail, what we do is we take an electrode, a glass electrode, and bring it down to touch onto an individual neuron. This glass electrode forms one half of the circuit. The other half of the circuit is dipped into the solution surrounding this particular neuron. If we zoom in, what we then want to do is form a really tight seal between this electrode and the cell membrane, the cell surface itself. And after we apply a short, sharp pulse of suction, we can break the cell just at the point of contact, allowing electrons to run and rush into the cell. They want to rush into the cell and back out towards the other end of the circuit, but they are prevented from doing so by the barrier in the way. And the only way they can get through are through these doors, through these gates known as channels. The only reason that these channels will open will be because of stimulus. And the only reason there would be stimulus there is if these cells are talking to one another. So we will receive and see electrical information when these cells are talking to one another. So I'm going to turn you all into electrophysiologists today and teach you one particular measure that we can make from this particular technique, and it's known as spontaneous excitatory postsynaptic currents. And this particular messaging system works like this. We normally have the electrical signal coming down to the boton, and then it causes the release of chemicals across this gap. When it's then received, it's then transduced and changed back into electrical signal. And I often liken this to a tap and a sink. We often have an electrical signal coming in and then a rush of neurotransmitter that will move across the gap and then received by the other neuron and taken away in terms of the signal. But what's important to understand is that in between those stimuluses and the rush of neurotransmitter, we actually have a leaky tap. And every so often we get a drip of the chemical moving from one side to the other. And when it does so, it provides stimulus for one of these gates, one of these channels, and it causes it to open and close very quickly. A short, sharp pulse of electrons then are able to rush out and go to the ground electrode, and we're able to measure them, which comes out as these spikes. Each one of these spikes is a quick opening and closing of one of these channels. And so what we're measuring is the spontaneous drip and the excitation of the channel, which is allowing the opening and closing in the post, the cell that's receiving the information, and the synapse is the structure as a whole, and the current that we're measuring. So the spontaneous excitatory postsynaptic current. And this particular recording can tell us a lot about how the cells are talking to one another. If we were to count up all of these different little spikes, which is the opening and closing of these channels within a given time period, we can work out the frequency, how often it opens and closes within a given time period. This frequency tells us the likelihood of the will for those cells to talk to one another, the propensity, the likelihood that they will release the chemicals in order to have a chat with one another. It could also be that there are more ears listening into the conversations. There's more synapses within this situation. In order to work out which one of these it is, we have to use a few other techniques. We have to fill the structure with dyes and count up the number of butons and the structures that are there to see if it has changed as a result of the measurement that we're making. We can get even more technical and use fluorescent stains that glow to tell us specifically about subsets of neurons that release specific chemicals. But in this way, this electrophysiological experiment allows us to understand how these neurons are talking to one another. So now we have what we want to measure in terms of the conversation that's going on with the cells, and we have the means in the form of this electrophysiological setup. But we need a model system to really understand what we want to see in terms of the progression of Parkinson's disease. Now, Parkinson's disease is actually for the most part, unknown as to the cause of it. We don't know whether or not it's familial or whether or not it's environmental. But there are a smaller subset, approximately 10% of which all Parkinson's disease has a genetic element that can be passed on. The normal genetic code within cells is normally deciphered by the cells in order to make proteins. And these proteins are like workers in a factory that each have their own job. They each go around and help the factory, the cell, work as a unit. Now, proteins can have a number of different functions which vary from breaking down fats to transporting other proteins to where they need to be. However, if we have a mutation, a change in the code or in the particular genes, we can actually result in a reduction in the ability for a protein to do its job, or the complete loss of a protein's ability to do its job, or indeed a gain of a new function which may indeed be toxic. Regardless, what we see is that about 10% of all Parkinson's cases have a mutation that changes the role of a protein and causes and goes on to contributing to Parkinson's disease. And one of the most regularly seen mutations in a gene and in a protein is in this leucine-rich repeat kinase 2, or LRRK2 for short, which I will refer to it as for the rest of this talk. Different mutations in this particular gene can go on to lead to Parkinson's disease. This large protein actually has various different mutations which can go on to a person or an individual developing Parkinson's disease in much the same way as the majority of cases, with the late onset and the similar clinical phenotypes and progression. This particular protein has lots of different sections to it. It's a very big protein and has lots of different sections to it. And each section is like a different tool. And the fact that it has so many sections and so many tools leads us to the idea that it has a lot of different jobs within the cell. And indeed, the data seems to indicate this is the case, that this particular protein within the cell has a lot of different jobs and roles. What's interesting is that a number of these different jobs revolves around this communication between cells, this chemical electrical activity that allows them to talk to one another. And so it seems that with this particular set of mutations in this particular gene, that we're led back to the communication between cells again. LRRK2's normal functions include changing the size of the synapse, assisting moving proteins that are vital to its normal function around the boutons where they release the chemicals. Indeed, it seems to be really important in releasing the chemicals that allow these particular neurons to talk to one another. So now we have a particular mutation or set of mutations in a particular gene which we can use as the basis for our model system. We can use electrophysiology to measure the connections between the neurons and see how they change between healthy neurons and LRRK2 mutation neurons. But which particular neuronal sites should we be looking at? Well, the answer to this might seem quite obvious. We have spoken already about the fact that the neurons of the substantia nigra pars compacta, these neurons that release that chemical known as dopamine, are quite preferentially destroyed in Parkinson's disease. And these particular neurons are found in an area of the brain known as the midbrain. They are situated under the bulk of the main part of the brain, just behind the little brain, which is the cerebellum, and just above the brainstem, which leads down into the spinal column. These neurons, these dopamine neurons, project and reach up into an area of the brain known as the basal ganglia. The basal ganglia is also another area of the brain that is damaged during Parkinson's disease. It is known as the lizard brain. It's one of the most ancient parts of the brain in terms of evolution, and is involved in things like survival. I need to find food, I need to forage for food, I found food, I need to remember this place because I found the food, it was great, that food tasted good, do it again tomorrow. But what's intriguing is that within these cells that die, the levels of the protein of LRRK2 are dwarfed by the amount of LRRK2 that's found in another area of the brain known as the cortex. The cortex, this upper part of the brain, is involved in higher processing. It's one of the things that separates us from other animals on the planet. If we take two brains on the right here, we can see that the cortex here is actually quite smooth in its structure. This is compared to a human brain, which isn't old and wrinkly here. It's a purposeful divots and folds that is here. If we think about it like this, if we have a set of neurons across this page, we're actually taking up quite a large surface area. But if we were to fold the paper, we're actually taking up far less amount of room, but with the same amount of neurons in the same space. So effectively what we're seeing here is these folds are space-saving techniques. They allow the brain to have a high computing capacity, but in a smaller area. It's much like the evolution of storage devices in terms of floppy disks from back in my day to what is now smaller micro SD cards. So this area of the brain, this cortex, also reaches out into the basal ganglia and helps form part of this particular circuit. And what we know is that when we have a circuit, it doesn't matter which part of the circuit is dysfunctional. We will stop the circuit from functioning in its entirety. So it doesn't matter where the disruption is, the whole circuit will suffer. So if we have a circuit in terms of the connections between the cortex, the dopamine neurons, and indeed the basal ganglia, if the issue is here in the cortex, we may be seeing a knock-on effect to these particular neurons because of their connection through this circuit. These neurons might be dying a bit like the canary in the cage. They might just be dying first because they're slightly more susceptible to this breakdown of the circuit. So we wanted to look at the cortical inputs into the basal ganglia. We wanted to look at these arms of these neurons in the cortex reaching down into this area of the brain that's damaged in Parkinson's disease. And we can do that in all three dimensions. We can look in the coronal sections, the crown sections. We can slice into the brain like this, or we can do in horizontal planes, or indeed sagittal planes. And we chose to look at in the coronal sections. This is so that we could keep the neurons in the cortex, shown here in the light brown, and the arms reaching down into the basal ganglia, shown here in this dark section, of which this is the striatum, which is the gateway into the basal ganglia. So now we have the means to look at the communication between the brain and the model system, and we know where and what we want to look at. So we went on to look at LRRK2 PD mutations in mouse models at very early stages. And what we found, measuring that drip, measuring that communication between neurons of the cell, is that when we compared it to the healthy neurons, we actually found that there was a significant increase in the communication between the cells of the brain. There seemed to be an increase in this drip, this desire to talk to one another within these particular brains at early stages. We went on to try and work out whether it was an increase in the desire to talk or whether or not there were more ears to listen by looking at the structures that were involved. We looked at the structures and saw no difference between the wild type and indeed the Parkinson's disease brains. We actually went on to look at it in that more fancy way of looking at specific connections and saw that there was no difference here either. And so what we were seeing was that there was a greater sense of connection between these cells. They wanted to talk to each other on a bigger scale, on an increased level. But all these changes can be quite abstract sometimes. What does this actually mean for the changes that we would see in terms of the motor behavior and indeed the non-motor behavior that we see in Parkinson's disease? Well, we did a number of behavioral tests, and one of those that we did was to look at the problem-solving skills, which I mentioned earlier. Problem-solving is impaired in Parkinson's disease. It's not as easy in people with Parkinson's disease. So one of the tests we did was to utilize something called the puzzle box. In this scenario, we put a mouse into an open area. And in this open area, mice don't often like to be in large open areas. They think that they're going to get swooped upon and taken away by a large bird and get predated upon. So what we did is we provided them with a goal box, which allowed them to find a nice, dark, safe, warm space. And the only thing preventing them from getting there was a wall with a small door. When they went through this area, we timed and stopped the timer to allow them to see whether or not how quickly they could reach this area. Mice are quite inquisitive, and so they would search around this open area until they found the door and entered into the goal box. We would then put them back into the arena a few more times to see how good their recall was, whether or not they remembered where the door to safety was. We would then make it a little bit harder by placing into the door sawdust so that they had to dig their way through. So now they had to remember where the door was and work out how to get through that door. So it was testing their problem-solving skills. We then took a Lego piece and put it into the doorway so that they then had to work out how to pull out the particular Lego piece, which they could do and before they could enter the goal box. And at every stage of this, we found that the Parkinson's disease models, shown here in pink, were far better at completing the task and remembering how to do the task than the non-transgenic littermates, than the wild-type control littermates. Now, this might seem quite confusing. What we've said so far is that there seems to be an increase in the connections between the brain in the Parkinson's models, and that there seems to be an increase in their ability to do problem solving. And this seems contrary and opposite to the normal state of play of what we're talking about in Parkinson's disease, where we see a decrease in the connectivity because we see neuronal loss, and indeed a decrease in the ability to do problem solving. So what's going on? It's important to remember what we're looking at here is not a disease of old age, but a model of aging and growing older. Normally what happens with neuronal activity over time is we'll get a progressive decrease in the amount of activity and neuronal activity with age. What we seem to be seeing in these particular models is that there seems to be a heightened level of activity within these particular neurons in the brain. What's interesting is that the body is usually quite good at keeping things within a happy medium. If we think about the temperature of our bodies being kept about 37 degrees, that's because it's optimal for all of our biological functions to operate in. But if we move a degree out of that either way, we begin to feel ill and the bodies don't function as well. So the body is very good at adaptation and compensation. And so if these cells are burning bright and are using up quite a lot of energy, it wants to rein it in and pull it in. And so this is what we hypothesize is happening. It will drag it back into a median state where it's happier. But if there's this constant, consistent push, this is going to be quite hard to fight against. It's like having an unruly dog against a leash that wants to run away and chase the squirrel. You have to bring it back into check. And if it stops and sits there, then it's fine. However, if it keeps pulling against you, eventually what might happen at one point is that the leash snaps and the dog runs off and tries to chase down the squirrel. We have a collapse of the system. So in this particular hypothesis, we might start off with an increased level of activity, but with time and with age, what we will see is that this fight against trying to draw this into a median is actually lost, and we see a collapse of the system. So if this hypothesis is true, what we should end up seeing is two phases to the disease. And if we remind ourselves, we actually saw an increase in that ability and that desire for the cells to talk to one another. And what we see at later stages is we are beginning to see data that shows that we're getting the opposite effect at later stages of disease. Now, this is all well and good, but what we really want to understand is whether or not all of these things are true in a human model. We want human cells to really have a look at this. And we are now able to look at human models and human cells for a human disease in the form of stem cells. We have now got the technology and the capability to take skin cells from individuals, often from skin biopsies that can be taken from anywhere, for instance, from underneath the arm, and we can drive them towards becoming stem cells. These stem cells have the potential to become any cell in the body. If we coax them in the right way, they can become skin cells again. If we coax them in another way, they can become ocular cells, cells for the eyes. And if we coax them in another way, they can become neurons. Now we have human neurons that have the same genetic makeup as the individual that they're taken from. And in this way, we can grow them on a dish. You can see here some neurons that we've taken from stem cells into neurons and how they project and are reaching out to the neurons around them in order to communicate. This here is our electrode. We are able to record from them and show that we can actually show up the electrical activity and the crosstalk between the cells. We can also drive them to bursts of activity, showing us even more about what is going on in terms of the cell's activity. So if we go on to measure that drip, that quantal drip in these particular cells, we can see that the LRRK2 neurons, the neurons that have this mutation that goes on to help develop Parkinson's disease, seems to have a decrease of activity in terms of that quantal drip. If we liken that onto the models that we showed earlier, what we see is that this seems to match up with the later stages of disease. So the human neurons taken from Parkinson's patients seems to mirror the later stages of our model where we have that collapse of the system. We are capable and have begun to look at in our preliminary evidences using these human stem cell models the ability to look at other aspects of excitability and the way in which neurons can communicate with one another. We're finding that the amount of input we need to put into a cell in order to fire bursts of activity is increased in the amount of Parkinson's disease. So in the Parkinson's disease neurons, there seems to be an increase in the amount of energy and excitation that we need to allow them to fire a burst. So it's actually harder for them to fire a burst of activity. Indeed, to get them to fire regularly, it also seems to be next to impossible. So there seems to be these issues in the normal ways in which these neurons communicate with one another. So these evidences are starting to show us data that is really useful in understanding that there is an element of miscommunication in the way that these neurons talk to each other, and that this starts and initiates much earlier than we first thought. And so we can begin to, if we can begin to understand that timeline, we can begin to interact and intervene at much earlier time points and prevent the breakdown of the cell. But this isn't the only way we can go about intervening and understanding how Parkinson's disease works. We know that there are other symptoms that precede the motor symptoms commonly associated with Parkinson's disease that initiate just before the diagnosis that we can intervene with. What I want to show you now is, with permissions, is a video from a person with Parkinson's disease, a gentleman called Tom Isaacs that's no longer with us, but was instrumental in co-founding Cure Parkinson's, which is a trust and a charitable trust that donates towards research that will help to find a cure in Parkinson's disease.
When you're diagnosed with Parkinson's, life changes beyond all recognition. Socially, it is a very visible condition, and yet to society it seems to be invisible. No one ever talks about it. It is, to many, a private misery. No one ever mentions
Apologies.
becoming frozen overnight in one position and the ongoing sleeplessness. No one ever talks about the side effects of the complex medication regime, which can be as extreme as the Parkinson's symptoms themselves. And no one ever talks about being imprisoned in your own body. And no one ever talks about the psychological changes that face people who live with this chronic condition. Parkinson's removes control. It destroys natural movement. It can be painful. Parkinson's changes everything in life that you used to take for granted.
So what we heard there from a person with Parkinson's disease is that it's not just a disorder of movement. It can affect other symptoms and impact other functions within the body that is just as important to those individuals. You can have issues with sleep, with anxiety, with pain, with problem-solving skills. And we know that these particular functions arrive and seem to manifest earlier than the motor symptoms. So this gives us a measurable point of intervention where we can try and stop the progression of the disease. So we want to learn more about and different types of non-motor symptoms, so in order to understand it better and see if we can intervene within this. So let's take one of these. Let's take sleep. We all know that sleep is a powerful factor in the daily lives. We've all felt so tired that it affects our emotional state, and it affects things like our coordination and our capacity to problem solve. That problem that is just presented to you just seems that much harder when you're exhausted. It can affect your motivation. And interestingly, all of these are symptoms that are present in Parkinson's. Sleep and the disturbance of sleep is one of the earliest signs of Parkinson's. The ability to get to, remain, and indeed have restful sleep is something that shows up fairly consistently in Parkinson's, in people with Parkinson's. Indeed, there are different sleep disorders which seem to convert at quite a high rate to Parkinson's disease, specifically REM sleep behavior disorder, REM sleep behavior disorder. This is a disorder which causes individuals to act out their dreams and actually, in some cases, violently act out their dreams and orate and vocalize their dreams to quite a high degree. This is an individual with one such sleep disorder, and you can see that they draw and they speak and they move quite a lot during their sleep. Now, RBD, rapid eye movement behavior disorder, can convert quite a high rate to Parkinson's disease and is a precursor for Parkinson's disease. But we know that sleep and its disruption is very much integral to Parkinson's disease at large. And we also know that sleep is very important to regulating synaptic activity. Without sleep, our synapses don't work very well, and indeed it leads to all of the issues that I mentioned earlier, the issues of trying to problem solve, our emotional state, our motivation, our anxiety levels. And it's this which seems to tie it all back to that communication between the cells. So the question is, can sleep, which is of Parkinson's disease itself, be possibly exacerbating and making the whole thing worse? Recent evidence has shown that induction of better sleeping patterns within model systems seems to actually reduce the signposts that are seen in Parkinson's disease. This aggregation, this collection and sticking together of proteins that form Lewy bodies seems to have actually decreased in Parkinson's when we increase the level of sleep. So does sleep present to us a particular area that is measurable and that's detectable that we can then intervene in and help people with Parkinson's disease to combat things like the non-motor cognitive disorders? Well, what we wanted to do was look at biological rhythms that occur over 24-hour periods. These are known as circadian rhythms, and they drive biological functions and features over a day, such as drive biological functions such as sleep. The sleep, it actually affects our periods of wakefulness and restfulness within a 24-hour period. And one of the cues, the drivers for this particular effect is light. Light cues actually affect the way in which our circadian rhythms are regulated. So we wanted to see whether or not we could use light as a tool to change the circadian rhythms, to change these biological rhythms, and see if we can affect some of the non-motor symptoms within our model system. So we took some mice and we introduced their daylight cycles, their day and night cycles in two different ways. We did an abrupt change in just turning on lights and having a quick, sudden, bright interface between dark and day and night. So a quick change between dark and light cycles. And then we did a much more natural, slow progression, gradual increase in the light that we saw. And what we found was between these two groups, we measured the mice's anxiety levels on something called an elevated zero maze. These mice, as I mentioned earlier, are inquisitive, so if they're placed on this area, they want to explore the whole thing. However, they're more anxious about entering areas where they could be more easily predated upon. However, what we can see here is that the time they took to enter these areas was much smaller when we looked at the gradual light changes. When we used the gradual light changes, it seemed to us that from this preliminary pieces of evidence that we could reduce the anxiety levels that were seen there. And so this is something that we want to pursue further. What I want to talk to you finally about today is how Parkinson's disease affects different groups. This is a really important thing to think about and a really important part of the information and scientific research. if we don't understand how Parkinson's disease shows up in different groups, we cannot easily diagnose it, and this can be a problem. If we take something that's probably quite well known and we look at heart attacks, we all know this trope of somebody clutching their chest on TV and having profuse sweating before perhaps collapsing on the floor. However, if we take two different groups, men versus women, these particular symptoms seem to only lend themselves to what is normally seen within male groups. Within females, heart attacks manifests quite differently. We can have dizzy spells and uncomfortable pains between the shoulder blades at the back. And understanding this difference can lead us to diagnosis correctly. Without it, and we may misdiagnose, which means we will not give the proper treatment and increase the benefit to the quality of life. And the exact same thing holds true for Parkinson's disease. For such a global phenomenon, so little is understood about the different ways in which Parkinson's disease can manifest. However, from what little we do know, we can see that there is cause for further investigation. It's shown that Afro-Caribbeans have a tendency to have reduced effectiveness with the typical drugs that are given to Parkinson's patients. Indeed, there seems to be a higher rate of non-motor impact on Afro-Caribbeans with Parkinson's disease. The mood changes and depression that can sometimes affect individuals with Parkinson's disease seems to be more prevalent in Chinese and Korean and Mexican and Peruvian populations than they are in other areas of the world. Indeed, with male individuals that suffer Parkinson's disease, they are more likely to suffer the cognitive non-motor symptoms than women with Parkinson's disease. So you can see that it's really important that we understand these differences so that we can diagnose correctly and improve the quality of life. But even more than this, if we can start to understand the differences, we can start to see what is exactly the same between these groups. And identifying what is the same between these groups will really allow us to understand what is at the crux and the heart of Parkinson's disease at large, and will allow us to treat it overall. And this is one of the things that we hope to begin to do within our own work in our own lab. We'll take LRRK2 and the different mutations, and the different mutations seem to lend themselves to different prevalences in different ethnic groups. So there'll be more of one mutation in LRRK2 in one ethnic group than in another. But if we look at all of these mutations, we can begin to start to see the common thread. And when we find common changes, we will be able to identify these as being the crux and at the heart of what's actually causing Parkinson's disease. Indeed, in our initial findings, what we seem to see is that this communication between the neurons in the brain seems to be one of those things that is consistently changed in our models of interest. So what I've hopefully shown you today is what we are hoping to achieve, which is to develop a timeline for the disorder with the hope that it will help us to develop a therapeutic. We know that Parkinson's disease starts with some kind of insult, whether it be genetic, familial, or some kind of environmental process. But as a result of that, we start to get changes in the way that the neurons communicate with one another. These changes in communication then lead to altered behavior, and this altered behavior then leads to the behavior that we see at large. However, there are still gaps in our knowledge and our understanding of what's going on and how this can contribute to the changes in the communication within the brain. And as we move forward with this and fill in the gaps, we can hope to understand not only what is going wrong, but when it's going wrong, allowing us to treat it and maximize the effectiveness of any therapeutics that we then develop, which will hopefully then prevent and stop the destruction of neurons in the brain and prevent the onset of Parkinson's disease. So with that, I would like to thank those members of my lab, Dr. Dina Fatala, Dr. Harry Harrison, and Dr. Shiva Campello, who are with me on this journey to help try to identify how to stop Parkinson's disease in its tracks, as well as my collaborators, both here and abroad, without whose help I would not be able to get through all of this large amounts of work, and through the funders for generously funding the work to look at the earliest stages of Parkinson's disease as a means of trying to combat the disorder. And to you all for listening. Thank you for listening.
Hey, thanks very much indeed, Dayne. That was absolutely fascinating, beautifully delivered with some great analogies and graphics. If our audience this evening would like to start to place their questions for Dayne, maybe in the Q&A, I'm sure Dayne would be very happy to take those questions. So Dayne, maybe if I could just start off going back to one of your earlier slides where you were showing those Lewy bodies in the cells that in the substantia nigra that produce the dopamine. And if I understood, that was as a result of aggregation of that protein alpha-synuclein, is it?
Correct.
Yeah.
Correct.
So is that protein unique to those cells, or why don't you get problems arising in other neurons?
It's a good question. Alpha-synuclein is a protein that its job is still being defined, but what we seem to see it being doing is actually helping to curve and stretch membranes. So it's actually involved quite likely in packaging up those chemicals that allows one neuron to talk to another one. The aggregation of alpha-synuclein, the sticking together and the clumping together to form these large lumps, is actually indicative of Parkinson's disease and does seem to happen within these dopamine neurons. Why it's specifically happening in these dopamine neurons, we still don't know, but it is seemingly specific to those. And so it's important that we do understand that, because it will give us some information as to why exactly these particular neurons are going wrong. So it's actually allowing us to understand the dysfunction that precedes that degeneration, because if we can think of it like this, it may well be that these huge lumps of protein have their own detrimental effect, or it could be that the fact that these proteins are coming out of solution means that they're not doing the job, so we're actually losing an effect. But either way, we're probably gathering up some kind of level of dysfunction because that protein is no longer doing the job that it ought to be doing. And so it's probably something specific to dopamine neurons. Dopamine neurons are actually quite large and are actually possibly the longest in terms, well, they are the longest in terms of their processes in their arms. And so it's probably to do with their energy consumption. Right.
Okay, excellent. Thank you for that. If I could maybe pick up a couple of questions that are starting to come in the chat then, Dave. Someone's asked about what overlap there might be between Parkinson's and other conditions.
Yeah, so it seems to be that it affects similar areas of the brain. The ataxia and the sort of rigidity is also affecting motor cortex, which is affected in Parkinson's disease, as well as dopaminergic neurons as well. So there is quite a lot of overlap. You can actually get induced changes in movement as a result of the drugs that are actually given for Parkinson's disease. So there is quite a lot of overlap between the two because of the regions that are actually affected.
Right, thank you. The person who asked that question is a founder of the ataxia patient group in Wales. Where in the brain was the excitability measured in the human iPSC study showing decreased ability of neurons to fire irregularly?
Yes. Sorry, I didn't mention that. So the neurons that we made in that particular study from the iPSC neurons, so from the stem cells, were actually generated and pushed towards a cortical input. So it mirrored what we were trying to do in our other models, which was to look at where the LRRK2 protein was actually higher. So we're actually looking at cortical neurons within the brain, so as to mirror that work.
Great, thank you. And to follow up on that, Dayne, how close are you to being able to differentiate those iPSCs into dopamine producing neurons? Is that something that can be achieved?
That's something that we have done now. So we are actually looking, we don't mean to discount looking at dopamine neurons. That's not the case at all. As part of a circuit, it's very important to look at all the neurons within the cell. So we have actually gone on to make dopaminergic neurons and found a number of changes in those dopaminergic neurons, as well as making neurons from the cortex also, and having seen changes there also. So we can make dopamine neurons in a dish. They are needing to be characterized fully, so that we actually understand all the pros and cons of using that particular model as a model for disease, because with all models there are pros and cons, and which is why we have to use all of them in unison to really understand what's going on, but that we can create them, yes.
And does that give scope down the road to maybe being able to transplant those?
It does indeed. It does. There is that possibility. I wouldn't say that we were at a stage where we would want to try that on a regular basis yet, because there are stumbling blocks in the way in generating neurons, but there have been tests done that have put these into model systems, and they have shown huge benefits. So yes, we are on the road to doing that, and there are certain labs that are using these as a way of actually utilizing the iPSC stem cell neurons as therapies in themselves, as opposed to models to just look at how the disease functions. So that is something that is being looked at by several groups, yes.
Yeah, very exciting, yeah. What do you think will be the greatest advancement, progression in treatment of Parkinson's disease in the next decade?
I think this is a good question. I think understanding the process of Parkinson's disease in its entirety is really going to lead us there. I think that the earlier and earlier we can identify changes and markers that mean that we will understand how Parkinson's disease is then being, how we're going down the road to Parkinson's disease, is critical. And so I think it's quite, one of the biggest things is the advent of this stem cell technology. It's really going to give us the ability to measure changes over time in a human system. So I think iPSC's technology and that platform is probably going to be one of the biggest advancements that we have in terms of research. It can also, as we were just talking about, also be used as a treatment in of itself. So if we move forwards with that, with the understanding of that and how we can actually develop that into a therapeutic, it not only provides us with a model platform, but also with an ability to fix it. So I think the understanding of how stem cells work and change into neurons, and the information that information gives us is the greatest advancement that we have within this decade.
Yeah. Thanks for that again, Dayne. As genetic mutations make up only 10% and sporadic Parkinson's disease cases make up around 90%, to what extent do you believe these genetic models using genes such as LRRK2 can tell us about the origins of disease?
I think it can tell us an awful lot. As I was trying to say there, but perhaps got lost a little, is that these mutations, there are two sets of mutations. We can either get autosomal dominant forms or autosomal recessive forms. The dominant forms cause late onset Parkinson's disease, whereas the recessive forms cause early onset. The early onset can be very aggressive and perhaps doesn't follow the normal path of progression as the majority of Parkinson's disease. But the late onset ones, of which LRRK2 mutations are, actually seems to follow the same disease course as Parkinson's disease. Will it give us all the answers? No, but that's why we want to look at multiple different mutations that will allow us to get a more whole, holistic look at Parkinson's disease. So whilst it only does represent a very small, it does give us huge insight into what is actually being affected in Parkinson's disease.
Great, thanks, Dayne. Following up on that, does any other animal suffer from Parkinson's or an equivalent?
No. We have other model systems, but it's usually us doing something to the genetics in order to look at that as a model system. So no.
There are no spontaneous disease models.
Not to our knowledge, but no.
Yeah. Would you mind explaining again the conclusion that was formed when seeing that the mice were less anxious when the lights were switched on gradually instead of suddenly?
So this is very, very preliminary data. This is something that we've only just started to do within the lab. The premise being is that we would want to change the cues that alter the regular biological rhythms within mammals that can then lead to changes in circadian rhythms and indeed, hopefully, sleep. What we tried to do was change the way in which they have a light-dark cycle and day and night within their home cages. And what we actually see is that if we do abrupt changes in light, so we just switch the lights on in the morning and switch them off at nighttime, we seem to have a more anxious phenotype in these animals. Whereas if we get this progressive shift in light intensity in the day and then the same at nighttime, it seems to produce less. This seems to be something to do with, and we are hypothesizing at this stage, modulating the circadian rhythm so they get better sleep. Some of the data that I didn't show you seems to show that they seem less active at the periods of the day where they should be the most active. So it seems to show that they are seemingly having a more progressive increase in the day and not the shock to their system with just a sudden light change. We have a lot of ways to go, but we're trying to modulate the circadian rhythm to alter their sleeping patterns, and that's what we think it's all down due to, that their sleeping patterns are being affected and they're getting more restful and gentle waking up and gentle going to sleep, which seems to be affecting their ability to have restful sleep and anxiety levels.
And you think that might have eventual application in man as well then?
I think very much so. There have been instances of using light therapy for a number of different disorders, with Parkinson's included, but we really need to work out the nuts and bolts of it. What frequency is needed of light? How long do we need the light? When do we give it? But this is something very much, I think, that could be applicable to humans very quickly. It's something that is non-invasive. It's non-drug related, and so if there was ways to test this because of the solid basis on research, it should be able to be introduced very quickly and could have an enormous benefit for the non-motor functions that are seen to affect people in such a huge way.
Yeah, that's great. Do you think study of the toxic agents that can lead to induced Parkinson's could help us understand the origin in the disease?
There's been a lot of work that's gone into looking at that. There's things like MPTP and other drugs that cause effectively destruction of certain areas of the brain called lesion models, and that has led us to understand key players because of the way that those toxic agents work. Because we understand what those toxic agents work on, for instance, mitochondria, they are the powerhouses of the cell. If we see the destruction of those, we now understand that mitochondria probably play a large part in Parkinson's disease at large. So these particular model agents and the work that has been done up to now has provided us with a large amount of information about the key critical players in Parkinson's disease. So I do think that they have their place in Parkinson's research. Yeah, 100%.
And do you give any credence to the idea that things like pesticides might be?
There's a lot of work that's been done with that, and there is there's researchers in the States, for instance, Beata Ritz, who looks into pesticides and environmental changes that can then lend itself. I think it could easily be that things like pesticides and other environmental factors could contribute, but it's likely to be a combination of genetic and environmental effects. The way that we ourselves process these toxic agents might be different between yourself and myself, and the slight change in the way that we process them might make you or I more susceptible to that having an impact on the system. So I do think that there will be there are important evidences that show that environmental factors are having an impact on developing Parkinson's disease, but I don't think it's as straightforward as just having the pesticides in our environment. There will be a genetic element too.
Thanks, Dayne. Are there resources that you would recommend for keeping up to date with advancements in the understanding of Parkinson's disease and its potential treatments for the future?
There's a whole host. Talks like this, I think, is an excellent resource in trying to keep up with what's going on in the terms of advancements in science as a whole. There are a number of talks that do go on in and around neuroscience. There are various societies that one can join. There are various groups. I am part of a particular group known as the World Parkinson's Congress, of which I'm a scientific member, and of which I have a mascot right here, which is Parky the raccoon, who is a mascot for the World Parkinson's Congress. The World Parkinson's Congress brings together individuals from all sides of Parkinson's disease, both researchers, medical professionals, as well as people with Parkinson's disease and carers, and a lot of that is put up online in terms of the lectures, where they meet every 3 years, and it's a wonderful way in which individuals from all sides of this particular fight against Parkinson's disease can get together, and individuals can join up and they can see all the talks online as well as get involved themselves. So there are a number of different societies, a number of different groups that can be and joined, and that person wants to email me, I can probably give them a list of different ways in order to connect with Parkinson's disease. But they are out there, and there is a number of ways that I do suggest you all try to keep up with Parkinson's disease research.
Excellent. Dayne, you seem to be very sensitive to striking a balance between giving hope to patients, but also not false expectations. Would you like to just comment upon that as well?
I think we should have hope. I am absolutely desperate to find out what's going on with Parkinson's disease, as are very many other researchers. I'm not alone in this, and there are fantastic researchers doing fantastic work. And I think that is one of the things that people should take heart in. This is why I think it's really important to actually talk and give more talks like this to tell individuals exactly what we're doing behind closed doors, so to speak, because I get to see the advancements in the research. I get to see the next step because I'm working in this area. And if you don't see that, you may well lose hope, but I can see the changes that are going on. And so I am hopeful for the future, but I know it will take time. And so I know that we're trying our hardest and we're moving as quickly as we can, but we want to get this right. We don't want to come up with a false positive. And so we want to end up with a situation that we can help everybody in the best possible way. So tempering it comes from a place of desperate desire to actually help, but understanding exactly what the state of play is within the laboratories and within research.
Great, thank you again. So I think we'll finish off with this last question for you, Dayne. I understand you've returned to your home city.
I have, yes. It's lovely to be back. It's lovely.
That's fantastic. And you're obviously a fantastic role model for young scientists, Dayne. And someone's asked, as a former career development fellow, how and why did you choose a career in neuroscience?
My career into neuroscience started a long time ago. I did my undergraduate at Leeds, and I went to Mayo Clinic, and I had the opportunity to work with people in the field. Mayo Clinic is a not-for-profit research facility, and I went into the laboratory of my former, and I would say my still mentor, Dr. Dennis Dixon, who had this fabulous desire for teaching and for neuroscience, and he would treat it as a teaching tool to really go down and sleuth out exactly what was going wrong in various neurodegenerations. And I've just really kept that going. I think that the brain is one of the most fascinating and interesting, as well as one of the most mysterious things that happens right here on Earth, right here within our own bodies. And I think it's really intriguing to try and understand what's going on. So I think it's because I've stayed in neuroscience because I don't think I'll ever lose the passion for trying to understand exactly what's going on. And I don't think the questions about how our brain works is going to dry up. So I think that's why I've stayed in neuroscience. There's so many different angles, so many different potentials, so many different questions that need answering that my particular brain won't let go. I can't let those questions just remain unanswered. So that's why I've stayed in it. There's such a vast plethora and wealth of areas to investigate.
Yeah. So Dayne, Parkinson's is obviously a disease that takes an enormous toll on those with the disease and those surrounding them. So we just wish you all the very best with your research program back here in Cardiff, and we're really proud to have you back here. So
Thank you very
I'd just
much.
like to bring in my colleague, Karen Edwards, for a moment, who's just going to capture some feedback for this evening.
Yeah,
Karen, you there? Great.
I am. Yes. Okay. Right. Can you see my screen?
And indeed.
Great. Okay. So those of you that have been before, you should know the drill by now. If you could go to menti.com and enter the code given here and just answer that first question for us. Where are you joining us from today? Okay. Just give it a chance while people can just submit their answers. Great. Okay. Cardiff, Wales, England. Fantastic. Okay, I'll just move along quickly while people catch up with that one. Thank you. So next question. Have you attended a Science and Health public lecture before? Okay, so yeah, a few have attended before, but lots of new people. So welcome and hope you enjoyed the lecture. Hope to see you at some of our future lectures too. Fantastic. Okay, so where did you find out about this evening's lecture? Lots of recommendations from teachers, colleagues. Great, some from our web pages. Lovely, thank you. Okay, so why did you attend the lecture this evening? You choose 2 reasons. Mostly to be informed and to be inspired, and I'm sure you will have been after Dayne's excellent talk. Okay. So as a result of viewing the lecture, great, most people feel their knowledge of the subject matter has improved, which is good. And people would like to find out some more and even undertake some further research. So it's very encouraging. And then how would you describe the evening's lecture in 3 words? Okay, great. Informative, engaging, inspirational. Excellent. Some great feedback there. Fantastic. Thank you. Okay. So I shall just stop my share and hand you back to James just to end off this evening and to tell you about our next lecture in the series.
Yes, it just remains for me to thank Dayne once again for a absolutely wonderful lecture this evening, and many thanks indeed, Dayne. The next lecture in our series will be Thursday, the 24th of March, when we have Professor Dame Teresa Martiaux from Cambridge University joining us, and her talk is entitled Changing Minds About Changing Behavior, 5 Ways to Better Health for All. So that's Thursday, 24th of March. Stay safe, everyone, and we look forward to seeing you again next month. Nostar, take care now. Bye-bye.
Nostar. Bye. Thank you.